Okay, here's the CME code. We got it a couple more times, so you'll catch it. Flu vaccination reminder. Another week or two to get this done. Please make sure it gets done so you don't get pulled from service. And the inaugural Duke Neurology Pickleball Tournament is coming up. Dylan, is this you? Who's doing? Felix? Okay, who's? Felix. This is Felix. All right. So, show up at Piney Wood Park, 400 East Woodcroft Parkway. See the email on October 8th for more details. Rich says he will be there. So go challenge Rich to some pickleball. And All-Stars, you may nominate. We have one All-Star this week. And this is why you're on my mind, Dylan, because I saw this earlier this morning. And maybe you need to tell us what the Pioneer Clerkship is.
Like a longitudinal med student experience.
A longitudinal med student experience. Okay, I've been away from the M2 curriculum for a bit too long, I guess. So John is part of this Pioneer Clerkship and saw one of Dr. Ryan's patients in the stroke clinic, comforted the patient and her husband throughout the interaction while also performing a thorough neuroexam without guidance that discovered subtle ataxic features. And the patient's family thought he was a resident or independent provider and referred to him as a natural while making sure to highlight his care. And presumably you have recruited him so he will stay with us for residency. Okay. Yes.
You don't get the students unless you're an acknowledged master teacher. So you don't know what the Pioneer is.
Rich is pointing out that Dr. Ryan's teaching skills have allowed him to receive these Pioneer students. He's recognized as one of our best teachers. All right, so the Neurology Research Roundup, Dr. Agashi was second author in this JCN study that looked at seizures secondary to encephaloceles and the utility of electrical source imaging in these cases. And the authors showed that although the source for abnormal activity localized close to the lesion, it was often not in the lesion itself for various reasons. And MEVMER is the code. And today I have the pleasure of introducing Dr. Raja. She's an assistant professor of neurology here and faculty director of the Duke Early Phase Research Unit and co-medical director of the Duke Research at Pickett Road Outpatient Facility. She graduated from Sidney Kimmel, formerly Jefferson Medical College, and then did a master's here, did a residency in Maryland, and then came here for back-to-back fellowships in neuromuscular and advanced neuromuscular. And today she will be talking to us about concepts in early clinical drug development.
Thank you, Dr. Spector. Good morning, everyone. Today's talk is really going to be focused on something that is not necessarily neurologic in origin, but it's a more conceptual and theoretical talk. So feel free to ask questions along the way and we'll get started. So what I wanted to do today is really focus on the part of drug development that occurs where you're in the lab and going all the way up to phase 1. We have a lot of experience with phase 2, phase 3 clinical trials within our patient populations, but what happens to actually get a drug to that point? So that's what I want to focus on today. Yeah. Is that better? Okay. So the overview of today's talk will be to review the goals of a traditional clinical development program and then kind of break it down into the preclinical development phase and the early clinical development phase. So to get started, we have to think about what actually is a drug. We prescribe drugs all the time, but what exactly is it? And so the FDA defines a drug as any substance that's intended to diagnose, cure, treat, mitigate, or prevent disease. It can also be defined as anything that affects the structure or function of the body. So what this means is that we have a huge variety of compounds, if you will, that could be considered drugs. We have our small molecules, which are the things that are generally made in a chemical fashion synthesis with production at scale very easily. And then we move into more complex products. And as we move into more complex products, we start getting into the class of drugs known as biologics. This is the class where we've really seen an explosion of products in the last few years. And this is a class of drugs that actually require a living organism in order to be synthesized. And so they're very sensitive to the production environment conditions. You're changing a medium, changing a cell line, changing anything, any very small element of it will have an impact on the final product. And because these are complex and internally heterogeneous structures, you have to really be very, very cautious about ensuring that you're going to have consistency in the production and manufacturing. This is one of the biggest challenges with this type of product and why development of this particular class of products is so challenging. And so the goal of clinical drug development overall is to basically make sure that we're getting the right drug to the right patient at the right dose at the right time. And so this can be a challenge in many cases. And when we think about this, prior to the development of genetic therapies and biologics, drug development was basically in the stage where you have a compound, you give it to a group of patients, some of them may benefit, some of them may not benefit. Now with the advent of biologics and use of diagnostic biomarkers, we're getting into more of an era in which you can fine tune and make a decision for which drug is actually going to have the most effect for which particular patient. And the place where we really see this is in oncology. And we're also seeing it now in the neurologic space in terms of some of our rarer genetic conditions as well as our neurodegenerative conditions. And so this is a long process. On average, it takes about 15 years to get a drug from get a drug through this process. And the average investment is on the order of $2.6 billion. And that's in 2013 dollars. So that's only going to have gone up with the rate of inflation. And if we think about that, that's actually an average. So the actual cost can be anywhere from about 115 million all the way up to $6 billion. And what's really going to determine the cost of development is going to be the type of disease process and the type of product. A biologic product will always be more expensive to develop because of the inherent challenges with the manufacturing process and ensuring consistency. And then if we think about this, less than 12% of the drugs that make it into phase 1, which would be where we're starting to see how this works in humans, less than 12% that actually get here make it all the way through the approval process. So this is not something that you would take on lightly, nor is it something that occurs quickly. And then the other challenge here is that, we said there's a lot of time and a lot of money involved. And so if you think about this, there's a lot of different stages. There's discovery, there's development, and there's manufacturing. A lot of this is funded by private industry, and a lot of it is also funded by government. About $22 billion comes annually from the NIH to support basic science research and clinical trials focused on drug development. And then you get $102 billion from industry. There is also a huge push for transfer of publicly funded research to universities into industry. And so that has made the process a little bit easier. What we also see is that government can fund industry. We saw this really in action during the COVID pandemic, where the government was funding development of therapeutics or of investigations to actually assess different therapeutics for treatment of COVID. And so why are clinical trials so expensive? And the reason is just this. As science has advanced, we've developed products that are more complex. We have a higher proportion of biologics to chemicals. We have increased trial complexity with development of adaptive designs or additional biomarkers that monitor response. Clinical trials are involving more patients. Technology has resulted in the need for more data sources as well. And then we're now venturing into therapeutic areas that may previously have been unattainable. We're looking at chronic disease. We're also looking at degenerative diseases. And neurology is one of the areas where there's been a real burst of drug development in the past few years, particularly with drugs coming into clinical trials as well as drugs coming into market. So based on what we have so far, we know it's going to be expensive. But the question is, what does it actually take? And so I like this slide because this kind of gives a nice visual representation of what actually is involved. And what you can see is that, we start in the pre-discovery stage where we're looking at clinical observations, we're asking clinical questions, we're doing basic science research to understand the mechanism of a disease. Then we're getting into the drug discovery and preclinical studies where we're identifying what our potential target might be. What kind of product are we going to use? Are we going to use a peptide? Are we going to use an antibody? Are we going to use a cell or gene therapy? Or is this going to be a vaccine? And only then, after we complete all of that, can we actually move into human clinical trials. So human clinical trials actually occur fairly late in the clinical development stage. And then we move into the review and approval process, and then there will often be post-marketing evaluation. We've seen this particularly with a lot of the newer neurologic drugs that have come out in the last few years, where they have significant surveillance post-marketing to assess for safety as well as long-term side effects. And so the question is, where do we start? So there's two general paradigms for this. One is to use a target-based approach, and the other is to use a phenotypic approach. In a target-based approach, what we're trying to do is really understand the pathophysiology, understand what's going on at the molecular level so that we can match a product to determine whether or not it's going to, quote unquote, fit into a specific receptor or have a particular effect. And so this is often done with technology, use of computer-aided design, and other forms of artificial intelligence. The other way that we can do this is with a phenotypic-based approach. And these are actually agnostic to the target. These were based more on you put something in to a system, and then you see what the effect is that comes out. And that's really what you're looking at. And so in order for this to work, you actually need a model system. It could be a cell, it could be a tissue, it could be an organism, and you're almost working backward. You're saying, okay, we know this works, but why does it work? And what is the part of it that is working? And so what do we have to do to actually identify a lead candidate and move it forward? And the key is that we need to understand, how does this particular compound or candidate relate to human disease? What do we know about the disease in its current state? What kind of physiology is impacted by the disease? Do we have an animal model that we can see, in which we can test this out and see, does it work? Does it produce results reliably? Or does it work across a number of different systems? Does it work in vitro? Does it work in tissue? Does it work in one organism? Does it work in another organism? And do we have a way to actually measure that in a standardized fashion? And then the other part that will come into play is, what are the biochemical properties of this drug? Is there going to be, is the drug specific for one particular receptor, one particular target? What is how well does it bind to that target? How well does it move through the body? And how potent is it? Can you actually achieve a reasonable level of this particular drug with a fairly small amount? Or does it take an amount that would be unreasonable? And so the preclinical work is really about preparing to test out the drug in humans. And so in that process, what we're going to do is really select those leads. We're going to optimize their properties. We're going to try to characterize the product as much as we can, looking at what it does to cardiac function, what it does to proteins. Any drug that it has significant protein binding is going to be problematic, because then you're dealing with a free circulating component and a bound component, which may impact how long the drug sticks around. You're dealing with metabolism. You want to make sure that you don't have significant CYP450 interactions, which could create problems with other drugs. You also will have to perform extensive animal toxicology studies to look at what the short-term effects are, as well as what the long-term effects are, depending on what the intent of the drug is. And then based on those animal toxicology studies and the clinical characterization, you will actually select your doses for the first in human study. The other key for this is that you have to be able to manufacture a quality and pure product that is going to be safe to administer to humans. This is often one of the stages where drug development really fails because there's not a way to actually scale up manufacturing. And so I want to take a minute to kind of talk about the difference between pharmacokinetics and pharmacodynamics, because this is really what that early and translational phase is all about. So pharmacokinetics is what the body does to a drug. And so you can think about this in terms of the absorption, the distribution, the metabolism, and the excretion of a drug. And then pharmacodynamics is what the drug does to the body. So what is the physiologic effect that we're looking for? And you can kind of think of this as, where is the drug having its action? How do we measure that action? What are we expecting to see? And this is all based on figuring out, is this going to be viable in humans? We want to ensure that we are going to have a good likelihood of success within a human and that it's actually going to work the way we want it to work before we move forward. And so just as an example, in pharmacokinetics, what we're trying to do is really characterize what the drug is doing over time inside the body. So we generally look at plasma concentration curves and time after dose to figure out, okay, how does this drug get distributed? How does it get eliminated? How long does it stick around? And this will also be dependent on the method of administration. Are we talking about a drug that's IV? Are we talking about a drug that's inhaled? Are we talking about a drug that you're going to take by mouth? Are we talking about a drug that's going to be given intrathecally? Whereas with pharmacodynamics, what we're doing is we're really looking at what is the effect. So in this example, you see you have a control brain showing activation of dopamine in the basal ganglia. In Parkinson's, you have reduced activation, but then after administration of the compound that is being evaluated, you now have increased activity of dopamine. This will be a pharmacodynamic marker. We don't know anything from this picture about the concentration of the drug in that tissue. And so the drug delivery system is really key here. And this is really important to us in the world of neurology because we're often trying to get penetration into the central nervous system. So there are some drugs that we have to administer intrathecally. Some of the newer drugs for spinal muscular atrophy, some of the drugs that are in development for Alzheimer's are great examples of these. Also, some drugs need to be administered IV, so then you don't have to worry about what's going to happen in the first pass metabolism after you swallow it and it goes through the liver and gets distributed. Is it going to be a drug that's inhaled? This has been a particularly attractive area in the world of epilepsy for treatment of acute seizure. And then kind of the traditional standby is your oral drug. So you really have to think about how you're going to administer the drug early in the development process, because that will guide your development long term. And really what you want to do is, as you move into clinical trials, you want to use the formulation that's actually going to be used at the end, because that is what you need to test. And so the route that you administer a drug and how you formulate it can really have an impact on the overall pharmacokinetics of a product. You could have a great drug that works really well in vitro, but how do you give it to an animal? If you give them, say, preparation A, it's toxic. If you give them preparation B, it's effective. If you give them preparation C, it's ineffective. You get detectable levels in all cases, but in the case of preparation A, you make the animal really sick. In the case of preparation B, you get what you want. And in the case of preparation C, you don't see any effect. So you're kind of trying to see how well does this move into humans? Do we expect the same kind of behavior? And so everyone has heard the term IND. IND is investigational new drug. So this is a drug that is essentially, this is kind of the permission, if you will, from the FDA, who is our regulatory authority in the US, to actually give a drug to humans. And in order to get to this stage, we actually have to demonstrate a number of different things. We have to demonstrate that it's going to be safe to give to humans, or at least that we have enough data to say, we can predict what some of the toxicities or some of the problems might be. We have to be able to actually manufacture it. We have to be able to we have to be able to monitor effectively and know that the individual who is going to be having oversight of the individual who's going to be having oversight is actually qualified to do so. And then there's many different types of investigational new drug applications. You have your commercial INDs. These are your traditional, most common IND where you have a pharmaceutical company or sponsor who is trying to push a product through development. You can have investigator-initiated INDs. These are often used for small-scale products where it may be something that's available on the market, but you're looking to use it in a different way in a particular group. Emergency use INDs were extremely common during the COVID-19 pandemic, where drugs were still in clinical trial, but there was authorization to go ahead and administer them to patients who needed them. And then you could have treatment INDs, which kind of you could break down into compassionate use or expanded access, which you can also think about as use of the drugs in a population that has not been formally studied. And so this is a kind of a busy slide. I present this only to provide an overview, but what I really want to focus on now is the part that I have highlighted in red. What actually happens in each of these phases? And so this is the Cliff Notes version. So we kind of talked about the preclinical phase, and now we are approved for testing in humans. In phase 1, we're going to look at a small number of participants, a small number of test subjects to see to really try to characterize the drug. And really what we're trying to do here is assess safety. Is this drug in this formulation going to be safe, and can we achieve levels that we think would be needed to have an effect? In phase 2, we're going to start introducing the product into patient populations and really saying, does it work? Do we see a signal for this product working? And phase 1 and phase 2 can overlap slightly, particularly depending on the therapeutic area. In some cases, it may not be ethical to actually do initial trials in healthy volunteers. A good example of this would be neurodegenerative diseases or oncology. You don't want to give a chemotherapeutic agent to somebody who is healthy because of the potential side effects. And so in many cases, initial safety studies can be performed in patient populations without a therapeutic intent. And so in phase 3, you're kind of doing a bigger version of your phase 2 to see how is this behaving in a larger group of patients? What are the kind of big side effects that you're expecting once you expose a larger group of patients to this particular drug? What are major safety concerns? And this can really drive the label and the use of a particular drug. And also, this is where you kind of can develop some of those black box warnings that come with a drug. And then only after you complete all of that can you actually ask the FDA to review everything and say, can we do this? But then even after the FDA reviews it and says, yes, you can market this, you're still obligated to do long-term safety and efficacy studies. And this is a part where many times we can see drugs being pulled from the market. We can see drugs that go through what's called an accelerated approval process, where they are approved conditionally on the basis that there's additional studies that demonstrate efficacy. So it's not necessarily a straightforward process. And so to distill down and talk more about phase 1, really, the objective of a phase 1 trial is to really characterize the pharmacokinetics. We want to know, how does the drug behave when it's in the body? What do we need to do to get it to steady state? How is it eliminated? Is it eliminated through urine? Is it eliminated through the gut? Is it eliminated through sweat? What have you? We also need to try to figure out, what's the optimal dose? How much? How often? How long do we treat? Is this something meant for chronic use, or is this something meant for acute use, like an antibiotic that might be on a 7 or 10-day course? And then the route of administration, as we discussed. We also really need to focus on the safety. Can we actually give this to a human without causing significant side effects within the body? Likewise, if we do cause side effects, are they so significant that nobody would ever want to take this? Or are they relatively mild and easily addressed? And then again, we want to see, is the drug actually behaving the way we want it to behave within the body, which is the pharmacodynamic piece. And so really the question becomes, why do we measure it? And the reason that we measure it is because we really want to know if we're getting to the right drug, the right patient, the right dose, and the right time. It's all about ensuring that we are going to do something that is not going to be harmful and has potential to offer benefit. And so what are the properties of an ideal drug? So ideally, we would want something that is going to be quickly absorbed, and that's going to be consistently absorbed. We don't want a drug that's going to be affected by a lot of food or pH-altering medications. The classic example of this is something like levothyroxine, which must be taken on an empty stomach for maximal absorption. The distribution is this: if we give X dose, can we predict that we're going to get Y rough concentration within the blood? You know, or we have a very narrow therapeutic window. For example, something like warfarin, where one patient may be fine on 2 milligrams and have a therapeutic INR, but someone may be super therapeutic on 2 milligrams of warfarin. And then we want to ensure that we're actually getting to the target quickly. We don't want to have to wait a long time for the drug to have an effect. And then we want to also make sure that it's not accumulating. We want to make sure that we don't have a depot of this drug that's stored somewhere in the body that may be released at some unspecified time point in a way that's unpredictable to us. And then we want to ensure that the drug is not extensively metabolized by the CYP450 system within the liver, because that is one of the major systems that sets up for drug-drug interactions. And then likewise, we want to ensure that the way that the drug leaves the body is not dependent on things like age, race, gender, comorbid diseases. Because these are all things that will impact the dose level or the dose interval at which you give something. And so I have here a figure that kind of describes what are the main parameters that we're looking for. So we're looking at the clearance, which I mentioned. We're looking at the volume of distribution. How is this distributed? Is this distributed in the plasma space? Is this distributed in the fat? Where is it distributed? What is the concentration that's achievable? What is the area under the curve? What is the total exposure to the drug? What is the half-life of the drug? And what is the time that it takes to actually get to the maximal concentration? And then how available is the drug? This gets down to, are you giving the drug IV, where you're going to immediately get a high concentration, or are you giving the drug by mouth, where you will expect to have a slower increase in the concentration over time? So other key things are that you want to make sure that you're not going to cause any cardiac dysfunction. QT interval prolongation is a big reason why drugs fail in early development, and a reason why so many clinical trials monitor ECG very carefully. While there can be some predictions that are made based on in vitro assays, you don't know for sure until you actually administer it. Likewise, especially in the world of biologics, you want to make sure that you're not having any immunogenicity. The body is not reacting in a negative way to the drug that you're giving. Similarly, there's no off-target effect. And you want to ensure that you don't have a narrow therapeutic index. So you want to make sure that it's a drug that ideally doesn't require a lot of therapeutic monitoring, or has a very predictable efficacy and safety so that you can use it regularly. And so I have some illustrations here to demonstrate kind of what you're trying to avoid. Is you're really trying to avoid these off-target effects, which would be elsewhere in the body, and really optimize your therapeutic window. You ideally want to have your therapeutic window be as wide as possible. And we think about therapeutic window many times when we're on the inpatient service and looking at what level of antibody do we have, or in the case of stenting, looking at the PFA activity levels. And so we hear the term, we need to get through phase 1. And what does that actually mean? Well, what are we doing in phase 1? The key is that we're establishing what the major safety issues are. And we're determining what we need to do to move forward into phase 2 and 3. What kind of dosing are we going to use? What kind of safety monitoring are we going to need? What kind of considerations do we need to make for other concomitant medications or comorbidities that would be common within the population of interest? And then really what we're trying to do is identify the knowledge gaps. What do we think based on the preclinical testing, and what is reality? And this is an area where we can often run into issues, especially if when we design a trial, if we are looking at a patient population that's too specific. Are you going to get that ideal patient population to do your study in? Probably not, because humans are not lab animals. And then the other thing that you're trying to do is figure out how can we determine early whether or not the drug is acting the way we want to do it. And you may not think about it, but phase 1 actually has a major impact on the drug label, the ultimate prescribing information that we are provided by the manufacturer to share with our patients. The dosage and administration, the dose forms and strengths, contraindications, warnings and precautions, interactions, specific populations, what happens when you overdose, the overall pharmacology, the pharmacodynamics, pharmacokinetics, counseling. These are all things that you're going to lay out and establish from your phase 1 studies. And so what is the most common phase 1 study? And this can be variable, and it's really directed by the goals of the study and the population. The most common forms are dose finding studies where we're trying to figure out what is the optimal dose, what is the maximum tolerated dose. And so these are typically conducted in healthy volunteers unless it would be unethical to conduct in a healthy volunteer. And so the single ascending dose study basically will use in a stepwise fashion administration of different dose levels, where you will have a dose that you're predicting based on your preclinical models will be accurate. And then you want to look at that dose as well as doses above and below to try to determine what's the maximum dose that you need. And then in a multiple ascending dose study, you're essentially trying to achieve steady state with your drug and figure out how is it behaving as you give it and how is it behaving as it ends, and really optimizing the interval at which the drug is given. There's other types of studies that can be done in phase 1, but these are the two that are really the key to moving forward and that will guide further testing. There's also crossover trials where you can look at food effects, bioavailability trials. This is particularly important if your initial testing is in a drug that is an IV route, but you're trying to make it an oral route. We see this a lot with antibiotics, where we have a different dosing paradigm for whether we're administering IV versus by mouth. Bioequivalence. This is particularly important when we're talking about a test product versus a reference product. And in the age of generic medications, this is really important. And then drug-drug interactions. And so what do we actually do? We are trying to figure out what is the effect of this drug on the human body. And so we have to think back to the preclinical development. What kinds of things were we noticing in the animals, and how would they translate into what we would see in a human? And in some cases, this could be informed by other drugs in the class as a class effect. In other cases, it could be a completely novel drug where we're really dependent on what did we see in the animal. And so some of the typical things that we'll monitor will be things like vital signs, clinical examination, and if we're seeing signs of particular involvement of one organ system over another, we could do more focused examinations of that particular system. In some drugs, there's a heightened concern for ocular toxicity. In studies like that, you may want to do more aggressive monitoring of ocular function and do more testing to look at fundus photos or retinal vasculature changes. Also, we will typically look at a standardized panel of lab values based on what we are expecting to happen and to assess the overall safety. Typically, we'll look at chemistry, hematology, coagulation, urine, depending on what type of method of delivery it is and where the mechanism of action is. This could include even looking at CSF. And then ECG, because as I mentioned, we want to make sure that we're not going to create any kind of unstable cardiac rhythm that would increase risk. And then adverse effects, are there any particular symptoms or conditions that patients report on a regular basis after receiving this drug? And so now we're ready to kind of move forward a little bit more. And we've kind of gotten through phase 1. And we're moving on into phase 2, where we're getting into more clinical trials, more patient populations, and really trying to see. And so what we have to remember, though, is that drug development is not always linear. I think this is an illustration of that, but in reality, if you look at this, it looks like a mess, really, if you think about all the different places where you could go back and forth. We think about it as being very standardized, but in reality, you can have studies going on at any different phase, and at any point, you can go back to the beginning and have to start all over again. And so with that in mind, the whole idea is, what can we do to really optimize the production and optimize the success rate? And the key is that we have to get ourselves out of this traditional mindset of we have to go in this order, this order, this order, this order. The mindset that we have to think about is, how can we establish very early that our drug is going to work? And if it's not going to work, we drop it and move on. So kind of think of this as a quick win, a fast fail, or a go, no go point, where we want to really see, okay, we are getting the effect we want at a level that's detectable, and then we move it forward quickly. And so with all of this being said, it's a long process, it's a costly process, and the probability of success is low. But that being said, there is still a need for this, and particularly now in the world of biologics, we are really seeing an explosion of drugs within this category. And so the last thought that I want to leave you with is, why would a drug not make it through the process? And this figure is a good illustration of this. The main reason that drugs fail once they get into clinical trials is because they are not safe or they don't work. The other consideration to this is the upfront investment. As we mentioned before, the overall cost of developing a drug includes not just the direct costs, but also the indirect costs. Everything that the pharmaceutical company has to invest, or that the developer has to invest in ensuring that they have the appropriate systems in place. So this can range anywhere from a few $100 million all the way up to $6 billion. And that's just a range. As drugs are getting more complex, and as we're moving into more cell-based therapies as well as gene therapies, what we're seeing is that the cost of development is increasing, and the cost of the final drug product is increasing commensurately as well. And so in the late stages, as I said, the reasons that things don't work is because while it might be safe to administer to a human, it doesn't work in the human the way you would expect it to work in the animal. So you don't have enough efficacy, or you cannot actually give a dose that will achieve a sufficient level to have an effect. And I also leave you with the thought of orphan drugs and cancer drugs, because these are really the kind of key for where development is moving. There's an increasing focus on orphan drugs and increased focus on cancer drugs. So cancer drugs were recently, FDA determined that any drug that goes through the accelerated approval process actually has to demonstrate long-term that it has safety and efficacy. And this is coming around to haunt a lot of pharmaceutical companies and resulting in products being pulled from the market. Other idea is orphan drugs. This is one of the darlings of gene therapy trials and cell-based therapies, where you're really trying to target a very specific population with a very specific personalized drug that is only going to be used by a very finite number of patients. And so I have some references here, but I will open it up for questions if anybody has any. And I think Matt Ludke had a question. I don't know how no, okay. Any questions, comments? Yeah.
Something I go to all the time. Why is the FDA so rigorous with drugs and so loose with devices?
It really varies because it depends on the risk of the device. Every device is categorized based on how much potential risk it poses. And so
why don't they require the same level of efficacy? Large studies.
So devices are also required to reach a certain barrier.
Do you agree that they're allowed to put in devices with 10,000 abilities that are approved they seem to get approved with studies of 50 people. As long as they don't do harm, obvious harm, they seem to get approved.
Yeah, I think this is one of the areas where now there's really starting to be more investigation at the level of the FDA and even at the level of the investigators, because this gets down to, is there an advantage to having multiple redundant products, or what advantage does one device give you over another? Do you need to have 15 different ways of doing the same thing? And so I think now they are starting to look at this in more detail, but we also have to bear in mind that the division that manages devices within the FDA is entirely separate from the division that manages drugs.
Last week, we had a lecture, and the device is approved to put into your down. This doctor is putting it everywhere else and getting paid. You couldn't possibly do that.
No, you couldn't. And I think part of this comes down to the fact that what populations are you targeting? Are you targeting, you're looking at a population that is refractory or difficult to treat, not responsive to medications that are currently available. And so that changes the calculus and changes the risk equation for what you would allow versus what you wouldn't allow.
But if you want to get a PMA, marketing approval, doing pivotal studies, you can get a device approval by the FDA. But if you don't have solid data, no one should use it. So it's kind of
You can't just hand out a bunch of pills to people and say, give them a shot and see what happens. What you can with devices.
But similar, there's some genetic drug you actually don't need to demonstrate efficacy. All you need to demonstrate PPE. Similar in the device market, all you need is demonstrate safety. So I think it's a little similar.
So I agree, the device market seems to be just safety. You don't have to demonstrate that.
Well,
You have
pardon?
Inputs for the audience that was
Oh.
just need some of it.
Yeah, so the question is, the discussion relates to the difference between getting a device approved versus getting a drug approved, and the idea that it seems like devices are more easily approved, but in reality, I don't know that you can really say that, because part of this depends on what do you expect the device to do. If you expect the device to have a therapeutic intent, as Dr. Fang was saying, you have to prove that it's going to have a therapeutic intent. Is the intent of the device to have, if the intent of the device is to have a particular effect, then you have to demonstrate that the device actually has that effect within a human population.
Do it in the context of clinicians using it. It's not like drugs, which have to go through this huge regulatory process before you give it to anybody for therapeutic.
I agree with that. I think that if you want the device approval process not as rigorous and the drug actually is a two rigorous settings, device often will be more.
So the comment is that
I would say it's less rigorous, but you can have a device approval without it. That no one going to use it. CMS not going to reimbursement.
exactly.
Equivalency is an issue.
Yeah, so as Dr. Fang is saying, there is a slightly different bar for approval of a device versus a drug, but ultimately the question becomes, how are you going to use it? Is this the right device for the right patient? And is it actually going to do what you want it to do? If you're trying to develop a device to assess arm function, is your device actually assessing arm function, or is it assessing something else? So this is where you get into the problem of how do you validate a device to do what it wants to do, in the same way that we get into the process of how do you validate a drug that is going to actually work on the target that you want it to work on. Yes, Dr. Fang.
So how, maybe you can elaborate a little bit, how as Neurology get access to the internet, and what is the overall pipeline, tutorials, how we can get access? We want to just get initially early driver trial, what will be the process?
Yeah, so Duke has a number of different processes in place for investigator-initiated studies, and many times you can actually propose an investigator-initiated study to look at a new indication for an existing drug, or if you are actually trying to develop a drug de novo. And there are a number of offices within Duke, specifically the Office of Regulatory Affairs and Quality, that can assist with development of an IND application, as well as a commercialization and translation office that helps to find additional ways to support product development. Yep. Yep. And once again, today's CME code. And if anybody has any questions, feel free to shoot me an email.